Okay, in previous part we talked about how bacteria they can achieve diversity through transformation, transduction, and conjugation. If you remember in conjugation, so imagine if we have imagine this is a bacteria, so this is like the chromosomal DNA, and then some of the bacteria they have this small circular DNA that we call it plasmid. So imagine this plasmid carries like the genes for antibiotic resistance. Then we have another bacteria, the recipient, that doesn't have this plasmid. It still has the genetic information, but like the chromosomal DNA, but it doesn't have the plasmid. So with the help of the pilus, so this plasmid can move or can be transfer to the new bacteria. So then this bacteria are going to become antibiotic resistance too, because now it has that antibiotic resistance gene. So sometimes when this plasmid that we call it F plasmid or fertility factor plasmid move to this new bacteria, so it can become part of their genetic information, so it can go and get integrated to their chromosomal DNA. So I just use a different colors that can show you. So for example, this plasmid go and become part of the genomic DNA. So then we call this cell HFR cells, or high frequency recombination cells. All right, now if this cell now want to transfer this information to a new bacteria, imagine it makes like a plasmid. I can show you here with different colors. So imagine this is our plasmid. Now when it exits from the previous like from that chromosomal DNA, so it's not a very precise excision, so it can carry like part of the genetic information of that bacteria too. So now we call this plasmid F prime plasmid that carries like some chromosomal DNA information as well. So now you can transfer this, imagine this bacteria can transfer this plasmid that carries part of that genetic information, chromosomal information of this bacteria. So with another conjugation, so it can transfer this plasmid to the new bacteria. So this F prime plasmid now can transfer to this recipient cells by conjugation, and then it can transfer this antibiotic resistance genes plus some of the genetic information of that bacteria to the new bacteria. So now you can see we have some recombination here, so it means we have some cells that they have some new genetic information that they didn't have before. So it's kind of like the diversity that the bacteria they don't have it like the eukaryotic cells, but by these small and simple procedures they can have some diversity. All right, so you can see it here nicely. So this is our recipient. This is our donor cells, so you can see the plasmid gonna go integrated into the host DNA, the chromosomal DNA, and then is we call this HFR cells. So now these cells can be a donor now, so it can transfer the genetic information to the new bacteria. but when they want to transfer this information, so it can carry some of the chromosomal DNA of the bacteria too. For example, they carry a gene, we call it like a lac gene, so that they can carry it from the previous bacteria to the new bacteria. So the next thing that can cause diversity in prokaryotic cells, we call it transposons, so or jumping genes. So these are the usually repetitive sequences that they have this ability to move from one location to another part of the DNA, or from one cell to another cells. And there are some enzymes that they call it transposase that they can help for this jumping and movement of these fragments of the DNA. So and you can see, for example, this is a transposon gene that you see it in red. Then this has this ability to move from one part of the DNA to another part. So imagine this is gene B. This is a gene that can produce a specific enzymes or a specific protein. When this transposon gonna move and jump to this gene, so it can disrupt the gene, so it can make this protein or this gene dysfunction. So it, imagine it can cause like interruption in production of that protein or that enzyme. So it can cause like for example cancer, or malfunction or dysfunction of like that cell. So you may think that transposons probably like a bad thing, but the main reason for them was the diversity, and you know how important is diversity in evolution. But sometimes it can cause dysfunction and interruption of the gene too. So the scientists actually discovered about transposon was an American genetics, Barbara McClintock, that she discovered it in maize or corn, and then she received a Nobel Prize for her discovery about these transposons, and that you can find it in prokaryotic cells as well as eukaryotic cells. All right, so this is the summary of mechanisms of the genetic diversity in prokaryotics. It's very important that you can learn about them and their differences: conjugation, transduction, transformation, and transposition, that and how their definitions and how they work. So the next topic is gene regulation. So we're gonna focus on prokaryotic cells, which is much more simple than eukaryotic cells in gene regulation. So we're gonna talk about like how the genes can turn on or turn off in prokaryotic cells. You know, depends on their environment, their condition. They have to express some genes, or they have to silence some genes. So we're gonna talk about how this process that can happen in bacteria. So usually the prokaryotic cells, their gene regulation happens at the transcription level, but in eukaryotic cells, the gene regulation can happen at transcription, or after transcription, or before translation, during translation, after translation. So it's much more complex than the prokaryotic cells. This is a good example. If you remember Serratia, so this is a bacteria at 37 degrees Celsius. It cannot make that pigmentation that you can find it at 25 degrees Celsius. So it depends on their environment. So at 37 degrees Celsius, it means that the genes that could produce this pink pigmentation are off, and the genes that are they can make this pink pigmentation, which is kind of they make it actually in a harsh environment. For example, at 25 degrees Celsius is not the optimum temperature for them, so then it means this gene is on, so it can be transcribed and then translated to make this pink pigment. So now see how this works. So this is a nice like picture for you guys, that you can see like the prokaryotic cells, the gene regulation usually happen at transcription level. So when we're gonna make like a messenger RNA out of DNA, but for eukaryotic cells, it can happen at transcription, post-transcription, translation, post-translation, or even it can happen not at the genetic just genetic level, so we call it epigenetics, which means beyond the genetics level, so that due to the environmental changes and the condition, so the expression of the genes can vary as well. So, but our main focus here is on prokaryotic cells. So prokaryotic cells, they have a structure that they call them operons. So the operons, they made of some genes that they are related together, so they work with together. For example, they make the same enzymes that are involved in the same pathway. Then they have a promoter, so that we're gonna, if you remember, we talked about it during the transcription, that the RNA polymerase can recognize the sequence in the promoter and can land there and start at the transcription. And then they have some regulatory genes that they usually can make repressors and activators, that we're going to talk about what are repressors and activators. That these repressors and activators, they can bind to the area that we call it operator. So this operator, you can find it usually upstream of that like group of the genes that they work with together. And and then we have some regulatory genes that you can see in this picture, and they usually can control the transcription of the genes. So the combination of promoter, operator, and the structural genes with together, they call them operons. So there are different kinds of operons. The first one, we call it repressor operons. So that the best example is tryptophan operon. So imagine we have several genes, tryptophan E, tryptophan D, C, B, and A. So these are the genes that they work with together to make like amino acid tryptophan. And you know, this is an amino acid that the bacteria they need it in order to grow. So they can make it themselves, or they can get it from their environment. So imagine that the bacteria doesn't have like enough tryptophan in their environment, so then they need to make this protein or this amino acid in order to survive. So at normal condition, like as default, this gene is on, so the RNA polymerase can attach to the promoter, and then so you can see it here. So can attach to the promoter, and then it can read the sequence and make the messenger RNA with the help of the RNA polymerase. So now imagine that the bacteria, we culture it in a media that has already tryptophan. So it has enough amount of tryptophan in that media. So bacteria is smart, so it's not going to spend the energy or their resources for something that is already available to them. So it needs to turn off the gene that can produce tryptophan for them because it's already in their media and in their culture. So now the tryptophan can attach to the to the repressors that is produced with the help of those regulatory genes. So then it gonna activate the repressor, and then the repressor gonna attach to the operator. Imagine that, for example, now the repressor is like a block that can block the RNA polymerase. So when the RNA polymerase cannot proceed and it cannot move forward, so it's not able to do the transcription. So when there is no transcription, there is not going to be any messenger RNA. When there is no messenger RNA, there is not going to be any translation or making tryptophan. So as default, these genes are on, so they can go through transcription and then translation and make the tryptophan. But when we have higher concentration of we have tryptophan, so high amount of tryptophan, so then we don't need to make more. So the bacteria evolutionary needs to save those energy. So it's gonna, the tryptophan in the media gonna activate the repressor. So the repressor gonna attach to the operator, and then they're gonna block the RNA polymerase. So when there is no RNA polymerase, there is no transcription. When there is no transcription, there is no translation. Then we have inducible operon. The best example is lac operon. So imagine this, there are some genes that are related in production of lactase, which is an enzyme that can break down lactose, the protein of milk. So as default, if there is no lactose or there is no milk in the environment of the bacteria, they don't need to make lactase. So it's waste of energy to produce this enzyme when there is no lactose in their environment. So as default, these genes are off. So there is no lactase production. So there is, this is the promoter, so there is a receptor that attached to the operator, so the RNA polymerase is not able to move on and the transcription is blocked. But imagine that we add the milk to the media, or we add like lactose to the environment of this bacteria. So now they have to make the enzyme to break down lactose and then use it as a source of energy for themselves. So in the presence of the lactose, so the lactose gonna attach to the repressor, so it's gonna act as an inducer, and then it's gonna move the repressor away from the operator, and now the operator is open, so the RNA polymerase is not blocked anymore, so it can easily do the transcription. So when then we make the messenger RNA, then with the help of the ribosomes, they can do the translation, and now they can make the lactase to be able to use the lactose in their environment. So as default, these inducible operon, they are off, but if you have imagine lactose or in their environment, so they need to turn on these genes to make the enzyme for using the lactose as a source of energy. Please make sure to watch these videos so that I always link on the slides, so that can give you a better visualize information that you can learn this material easier. So this is also a nice slide for you guys that you can compare the gene expression of prokaryotic cells versus eukaryotic cells. So there are some other ways that the prokaryotic cells they can do the gene regulation. So one, we call it global responses, that we know it as alarmins. So these are like small intracellular nucleotide derivatives, so that they actually can change the gene expression, usually based on the environment that they have. If for example the bacteria is in a very harsh environment, so it is like alarmants, they can turn on and turn off the genes, and they can act quickly that to make sure the bacteria can survive in that condition by turning on or off the genes. So another type of global responses, there are attenuations and ribose switches. So they can control the transcription and translation, and this is how they can turn on and turn off the genes. For example, attenuation, this is where they can make like secondary stem loop structures within the 5' end of the messenger RNA that being transcribed, and this is how they can determine if the transcription can be completed in order to be able to do the translation or not. Also, ribose switches, they are also part of the 5' end of some prokaryotic messenger RNAs, and then they can cause the production of the stem loops, and these loops they can determine the completion of the messenger RNA synthesis and then protein synthesis or not. Then we have some enhancers. They can as you can imagine, increase the gene expression, depends on their condition. If they need like expression of some genes increases at a certain time, so this enhancer is gonna help. And then epigenetic regulation, so this is changes that happen for example by adding some chemicals such as the methylation. So you're gonna add like a methylene to the certain part of the DNA, and this methylation gonna cause like that part of the DNA turn off, so it can be like not anymore accessible for RNA polymerase to do like the transcription, and then when there is no transcription there is not going to be translation. So there is still some regulation that can happen even beyond the changing the genetic information, so that we call it epigenetic, and it's very common in eukaryotic cells. Also it can happen in prokaryotic cells as well. So then we're gonna continue with chapter 12, which is about the modern application of the microbial genetics. Thank you, and talk to you soon.